Welcome back, everyone, to the MedPod for another deep dive.
Yes.
Last time we were exploring atoms and the duality of light and electrons and even quantum mechanics.
Right.
But today we're going to take all that and see how it actually applies to biophysics.
Yeah, it's really fascinating to see how these fundamental principles of the universe actually play out in living systems
Yeah.
and impact your health and medical technology we use every day.
Yeah, you might be surprised to learn just how much these tiny atoms and their behaviors influence things like medical imaging or how our body functions at the
Exactly.
most basic level.
So let's start by building on what we discussed about atomic structure,
Okay.
specifically Bohr's model.
Oh, yeah, I remember that.
If you recall, describes electrons orbiting the nucleus
Uh-huh.
in specific energy levels.
Yeah, like tiny planets whizzing around a sun.
Yeah.
But a very specific path.
Exactly. And it's not just a pretty picture, though.
Right.
This model has real-world consequences.
Okay.
For example, it explains phenomena like line spectra.
Okay.
Think of the colorful glow of neon signs
Yeah.
that comes from electrons in those neon atoms,
Okay.
jumping between energy levels and emitting light at very specific wavelengths.
So each element has its own unique light signature based on how its electrons are arranged.
Precisely. And this principle applies to all atoms, not just neon.
Wow.
And this is the foundation of spectroscopy,
Okay.
which is a powerful tool used in medicine to analyze the composition of substances.
Okay.
So by looking at the specific wavelengths of light absorbed or emitted by a sample, we can identify the elements and molecules present even in tiny amounts.
So we're talking about using light to basically fingerprint the building blocks of matter.
Exactly.
That's
Yeah.
incredible.
Yeah, it's pretty amazing.
But how do we actually know that Bohr was right about those energy levels?
Right, so there was this ingenious experiment by Frank and Hertz,
Okay.
and they put Bohr's theory to the test.
They used mercury vapor and found that electrons could only transfer energy to the mercury atoms in specific quantized amounts,
Okay.
corresponding to those jumps between energy levels.
So it wasn't just a model.
Exactly.
It was like an actual thing.
It was a landmark discovery
Yeah.
that solidified our understanding of atomic behavior and paved the way for countless technological advancements,
Well.
including in the medical field.
So speaking of medical advancements, this brings us to X-rays,
Yes.
which we use every day for diagnosis and treatment.
Absolutely.
So I'm guessing those are also connected to these fundamental atomic principles.
Absolutely. Remember how we talked about the dual nature of light and electrons?
Yeah.
Well, X-rays are a perfect example of this.
Okay.
They are electromagnetic waves, just like light,
Okay.
but with much higher frequency and energy.
And that higher energy is what allows X-rays to penetrate our bodies
Precise.
to create those images we see.
Exactly. And it's all thanks to the clever design of an X-ray tube. Inside, you have a cathode that emits electrons, which are then accelerated by a high voltage towards a metal target called the anode.
Right.
When those electrons slam into the target, X-rays are produced.
Okay, so I'm picturing these electrons like tiny bullets being fired at a metal sheet.
Yeah, a good analogy.
What happens when they hit?
So there are actually two ways X-rays are produced in this process.
Okay.
One is called braking radiation,
Okay.
and this is when the electrons suddenly decelerate upon hitting the target, releasing their energy as X-rays with a range of energies.
Okay.
And then there's characteristic radiation,
Uh-huh.
which happens when the incoming electrons knock out electrons from the inner shells of the target atoms.
Oh.
When those gaps are filled by other electrons,
Yeah.
they emit X-rays with very specific energies.
Okay, that makes sense. But I imagine we don't just blast patients with all those different X-ray energies.
Right, you're absolutely right.
There must be a way to control it.
And that's where the concept of cutoff wavelength comes in. It's the minimum wavelength and therefore the maximum energy of the X-rays produced.
Okay.
By carefully controlling the voltage in the X-ray tube,
Uh-huh.
we can ensure that the X-rays used have the right energy to penetrate tissues effectively,
Right.
but also minimize any unnecessary radiation exposure.
So it's about finding that sweet spot, balancing clarity of the image with the safety of the patient.
Exactly.
Yeah, makes sense. And there are even more nuances to consider,
Okay.
like how different tissues absorb X-rays to varying degrees,
Oh.
which creates the contrast we see in the images.
Uh-huh.
So denser tissues like bones appear white because they absorb more X-rays,
Okay.
while softer tissues like muscles appear darker because they absorb less.
So the image is like a map of how much the X-rays are being absorbed as they pass through our body.
Exactly.
That's interesting.
And to further enhance the visibility of certain structures, doctors often use contrast materials,
Okay.
which are substances that absorb X-rays much more strongly than surrounding tissues.
For example, barium is commonly used for imaging the gastrointestinal tract.
So it's like highlighting specific areas of interest on the X-ray map.
Exactly.
Clever. Okay, so we've seen how atomic structure and light's dual nature are crucial for X-rays.
Right.
Are there any other key concepts we should touch upon before moving on?
Well, you mentioned gas laws and thermodynamics earlier.
Yeah.
They might not seem as directly connected to atoms,
Yeah.
but they're essential for understanding a range of biological processes.
Right. Those were topics we briefly touched upon in the previous episode.
Yes.
Could you refresh our memory on how they apply to the human body?
Of course, imagine your lungs, for example.
Okay.
The way we breathe, the exchange of oxygen and carbon dioxide,
Uh-huh.
it all relies on the principles of gas laws,
Mhm.
especially how the pressure and volume of a gas are related.
So when we inhale, our lungs expand.
Yes.
The volume increases, and the pressure inside decreases, drawing air in.
Precisely.
And when we exhale, the opposite happens.
Exactly. And this constant exchange of gases driven by these pressure differences is crucial for maintaining the right balance of oxygen in our blood
Okay.
and removing carbon dioxide.
So it's like our bodies are finely tuned machines
Yeah,
designed to optimize this gas exchange using the basic laws of physics.
exactly.
Amazing. But what about thermodynamics?
Right.
Where does that come into play?
So thermodynamics is all about energy and how it's transformed in systems.
Okay.
In our bodies, we're constantly converting chemical energy from food into mechanical energy for movement, electrical energy for nerve impulses, and heat to maintain our body temperature.
So it's like our bodies are running their own power plants,
Yeah,
governed by the laws of energy conversion.
exactly.
Fascinating. This is all so incredibly intricate and interconnected,
It is.
from those tiny atoms and their quantum behavior to the air we breathe and the energy that powers our cells.
It really is remarkable.
It's a symphony of physics playing out within us.
And we've only just begun to scratch the surface of this fascinating world.
I know, right?
There's so much more to discover, and in the next episode, we'll be diving deeper into the realm of proteins.
Ooh.
Those workhorses of our cells and how they create movement, transport substances, and even control the electrical signals that govern our bodies.
Sounds intriguing. I'm definitely looking forward to that. But for now, we'll take a break. Stay tuned because there's a lot more to uncover in our deep dive into the fascinating world of biophysics.
Yes, definitely. Welcome back. Before the break, we were discussing how these principles of physics are at play in some pretty amazing ways within our bodies.
Well.
But now let's zoom in even further
Okay.
and explore the fascinating world of proteins.
Yeah, proteins, they're often called the building blocks of life.
Exactly.
And for good reason.
Yeah.
They're involved in virtually every process that keeps us alive and functioning.
They're like the tiny machines that build and operate everything in our bodies.
Wow.
They catalyze reactions, transport molecules, provide structural support, and so much more.
So much.
And it all starts with their unique structure.
Okay, let's break down this structure then. What exactly determines the shape of a protein and how it works?
So proteins are made up of chains of amino acids.
Okay.
And the sequence of those amino acids is
Yeah.
what we call the primary structure.
Oh.
Think of it like the blueprint that dictates everything else.
So just like the order of letters in a word determines its meaning,
Yeah,
the order of amino acids determines what the protein is and what it does.
exactly. But it's not just the linear sequence that matters.
Okay.
That chain of amino acids then folds into specific 3D shapes, forming secondary structures like alpha helices and beta sheets,
Okay.
which are held together by hydrogen bonds.
Wait, hydrogen bonds. Those are those weak attractions between slightly positive and negative regions of molecules, right?
That's right.
I remember we talked about them last time.
Exactly. And while individually weak, they play a crucial role in stabilizing the protein's structure.
Okay.
And it gets even more interesting. These secondary structures then fold even further, creating the complex 3D shape of the protein, which is its tertiary structure.
Oh, wow. So it's like origami folding a simple sheet of paper into an incredibly complex sculpture.
A perfect analogy, and this intricate shape is absolutely crucial for the protein's function.
Okay.
It allows it to interact with other molecules in a very specific way, kind of like a key fitting into a lock.
Oh, so if the shape's wrong, the key won't fit and the protein won't be able to do its job.
Exactly. And to add another layer of complexity,
Okay, there's more.
sometimes you have multiple protein chains coming together to form even larger structures
Wow.
called quaternary structure.
It's mind-boggling how all these levels of structure work together to create such complexity.
It is.
But how does a protein know how to fold into the correct shape in the first place?
Right.
Is there some sort of instruction manual hidden in our DNA?
That's the million-dollar question that has fascinated scientists for decades.
Yeah.
It's called the protein folding problem. And while we've learned a lot, there are still mysteries to unravel. One of the most captivating experiments that shed light on protein folding was done by Christian Anfinsen.
Oh, what did he do?
So Anfinsen took an enzyme called ribonuclease A
Okay.
and unfolded it, basically disrupting its structure.
Okay.
But when he removed the denaturing agents, the enzyme miraculously folded back into its original shape and incredibly regained its activity.
Oh, wow. So it's like crumpling up a piece of paper and then watching it magically smooth itself out.
Exactly.
That's incredible.
It is. This experiment suggested that the information for a protein's correct 3D structure is encoded within its amino acid sequence.
Okay.
But then we have something called Levinthal's paradox, which throws a wrench into things.
Levinthal's paradox. That sounds intriguing. What is it?
Well, Levinthal calculated that if a protein tried to fold randomly by trying every possible conformation, it would take longer than the age of the universe to find the correct structure.
I had a really long time to get ready in the morning.
Indeed. And yet proteins fold in seconds or even milliseconds.
Yeah.
Clearly, there's something more going on than just random trial and error.
So it's like trying to find a specific grain of sand on a beach by picking them up one by one.
Exactly.
Utterly impossible.
Exactly. That's why scientists came up with the folding funnel theory,
Okay.
which suggests that proteins don't sample every possible shape, but instead follow a more directed pathway, guided by their amino acid sequence and interactions with the surrounding environment.
So it's like having a map that helps you quickly find the correct destination.
Exactly.
Okay, that makes a lot more sense. But what happens when things go wrong and a protein misfolds? Is that a problem?
Misfolded proteins can cause serious issues.
Oh, no.
And are implicated in many diseases, from Alzheimer's to Parkinson's to cystic fibrosis and even cancer.
Oh, wow.
It's like having a malfunctioning robot wreaking havoc inside our cells.
So how do our bodies prevent that?
So our cells have evolved special chaperone proteins that act like guides, helping other proteins fold correctly and preventing them from getting stuck in misfolded states.
So it's a highly regulated process.
It is.
That's reassuring.
Well,
Okay, so we've talked about the structure of proteins and how they fold.
Right.
How does this all tie into the concept of movement?
Right.
We've been calling proteins tiny machines, but we haven't seen them in action yet.
That's where motor proteins come into play.
Okay.
These are a special class of proteins that use energy to move along cytoskeletal filaments, like tiny molecular tracks within our cells.
Okay.
They're responsible for things like muscle contraction and transporting cargo around the cell.
So it's like a train running on rails carrying goods from one part of the cell to another.
A great image, but how do they actually move? What's the mechanism behind it?
Yeah.
It's a fascinating process involving a cycle of binding conformational changes and energy release.
Okay.
A great example is myosin, the protein responsible for muscle contraction. Myosin has a head domain that binds to actin filaments, which are a part of the muscle fiber's cytoskeleton.
Okay, I'm picturing it. So myosin grabs onto actin and then what?
When myosin binds to a molecule called ATP, it undergoes a shape change, kind of like cocking back a lever arm.
Okay.
Then when ATP is broken down, releasing energy, that lever arm swings forward, pulling the actin filament along.
So it's like a tiny ratchet pulling the muscle fiber bit by bit.
A perfect analogy. And this cycle repeats with myosin, detaching, rebinding to actin, and pulling, again, driving the contraction of the muscle fiber.
So it's a continuous cycle of bind, pull, release, repeat, all powered by ATP, the energy currency of the cell.
Exactly.
It's incredible how something so small can generate such powerful forces.
It is a testament to the elegant design of biological systems, and this process is happening constantly throughout your body, enabling you to move, breathe, and even think.
Wow. It's amazing to think about all this intricate molecular choreography happening inside us at this very moment. But before we get too lost in the world of proteins, we need to move on to our last section. Welcome back to the MedPod. We've been on quite a journey today exploring the fascinating world of proteins and how their intricate structures and movements are essential for life. But how does all this molecular action translate into something we can actually experience, like muscle function?
It's remarkable how those tiny molecular interactions we've been discussing can generate the forces that allow us to move, walk, run, and lift weights. It all comes down to the elegant design of our muscles.
Okay, I'm ready to delve into the world of muscles. Can you give us a quick overview of how they're structured?
Muscles are incredibly complex structures. Essentially, biological engines that convert chemical energy into mechanical work. And just like any well-designed engine, they have a hierarchical structure.
So there are different levels of organization within a muscle.
Exactly. Let's take a skeletal muscle like your bicep as an example. The whole muscle is made up of bundles of muscle fibers, kind of like strands in a rope. Each muscle fiber is actually a single long cell containing multiple nuclei. And within each muscle fiber are myofibrils, the contractile units of the muscle.
So it's like a Russian nesting doll with smaller structures nested within larger ones.
A perfect analogy. And the key player in muscle contraction is the sarcomere, the basic unit of the myofibril. It's like a tiny compartment containing highly organized arrays of protein filaments, primarily actin and myosin.
Ah, actin and myosin. Those are the same proteins we were just discussing, the ones involved in that ratchet-like movement, right?
Precisely. In the sarcomere, actin filaments form thin filaments, while myosin molecules assemble into thick filaments. These filaments are arranged in a beautiful overlapping pattern, giving skeletal muscle its characteristic striated appearance.
Okay, I'm starting to visualize it. But how do these filaments actually interact to make a muscle contract?
That's where the sliding filament model comes in, one of the most fundamental concepts in muscle physiology. Essentially, when a muscle receives a signal to contract, those myosin heads bind to the actin filaments, forming what we call cross bridges. Then the myosin heads pivot, pulling the actin filaments towards the center of the sarcomere.
So it's like a...
Exactly. And this braking system is controlled by calcium ions. When a nerve impulse signals the muscle to contract, calcium ions are released and bind to troponin. This causes a shape change in tropomyosin, moving it out of the way and exposing the binding sites on actin for myosin to attach.
So calcium ions are like the key that unlocks the braking system, allowing the muscle contraction to happen.
Precisely. And when the nerve impulse stops, those calcium ions are pumped back into storage, the braking system re-engages, and the muscle relaxes.
This is all so incredibly coordinated. But we've been talking about these nerve impulses that trigger muscle contraction.
Right.
Where do those signals come from?
That's where we enter the realm of the nervous system. Our brains and spinal cord constantly send out electrical signals that travel along nerves like wires carrying messages throughout the body. These signals are called action potentials.
Action potentials. That sounds familiar. Didn't we touch upon that concept in a previous episode?
We did. Action potentials are rapid changes in the electrical potential across a nerve cell's membrane. Think of it like a tiny electrical spark that travels down the nerve fiber.
Okay. I remember we talked about cell membranes acting like barriers. Could
Yeah.
you remind me how that relates to electrical potential?
Of course. Every cell in our body is enclosed by a cell membrane, which separates the inside of the cell from the outside environment. This membrane is selectively permeable, meaning it controls which substances can pass through.
Right, like a bouncer at a nightclub carefully selecting who gets in.
Exactly. Now imagine there are more positively charged ions outside the cell than inside. This creates an electrical potential difference across the membrane, like a tiny battery.
Okay, so there's this electrical tension across the membrane. How does that create an action potential?
When a nerve cell receives a signal, specialized protein channels in the membrane open, allowing ions to flow across. This movement of ions changes the electrical potential, creating a wave of depolarization that travels down the nerve cell's axon.
So it's like a chain reaction with each section of the nerve cell triggering the next.
Precisely. And this wave of depolarization is the action potential. It's a rapid all-or-nothing event, like flicking a light switch on and off.
So this action potential travels down the nerve cell and eventually reaches a muscle fiber. What happens then?
When the action potential reaches the end of the nerve cell, it triggers the release of neurotransmitters, which are chemical messengers that bind to receptors on the muscle fiber's membrane.
So it's like a relay race with the electrical signal passing the baton to a chemical messenger.
A great way to think about it. And these neurotransmitters then trigger the release of those calcium ions inside the muscle fiber, which, as we discussed, initiates the sliding filament mechanism and muscle contraction.
Wow, so it's a whole chain of events from electrical signals to chemical messengers to molecular movements, all working together to create a simple muscle contraction.
It truly is a remarkable symphony of biological processes, all orchestrated by the fundamental principles of physics we've been discussing.
It's mind-blowing to think about all this happening inside our bodies, every second of every day, without us even being aware of it. We've covered so much ground today, from atoms to action potentials, and I hope you've gained a deeper appreciation for the incredible world of biophysics and how it relates to your own body.
It's been a fascinating exploration, and I hope our listeners are inspired to learn even more about the intricate workings of life.
And speaking of learning more, I'm already excited for our next deep dive. We'll be exploring the fascinating world of cell membranes in even greater detail, and delving into the intricacies of action potentials and how they govern so many processes in our bodies.
It's a journey into the electrical language of our nervous system, and I can't wait to share more about these amazing mechanisms.
Until next time, keep those neurons firing and keep exploring the fascinating world around you.
Thanks for joining us on the MedPod.